Hi and welcome to the channel. If you don't know me already, my name is Salem Clemens. Been in the hobby for about a decade now. Been growing and selling corals for the last 5 years. And for the last 2 years, I've been working with my university to describe the full pathogenesis of various illnesses in the hobby. So STN, RTN, brown jelly, ECS. I've been getting a lot of good data on that. I often get asked to help people, and all of my solutions are rooted in science. I'm constantly reading peer-reviewed articles about the latest, new cutting-edge techniques to study corals, things they discovered about corals, and I typically filter all of my view of reefing through those articles that I'm reading. A lot of people have asked me if I could send them articles, and I certainly can, and if you'd like to be sent articles, let me know. But I thought a more direct approach would be bridging the gap between academia and the hobby by directly bringing that information to all of you in a digestible piece of media, a video. So I decided to make a YouTube channel. This is my first YouTube video, so I'm new to it. So any tips or help are appreciated. Hope to get a better mic, that's step one. But the ultimate goal of this hobby is to promote those science-based solutions and to help break down some of the more advanced and far out there topics that are being covered in science but aren't really being talked about at the hobbyist level. So because of the nature of the content I'd like to cover, this is going to be a little bit more advanced and not as beginner friendly, but hopefully with the structure of this video and the structure of videos going forward as I continue to improve, I can make this channel and the content I create something that everyone at every level of the hobby can enjoy. So the way I'll structure this content is I'll start out with basically the bare bones, the most basic side of the concept, and then as the video progresses, I'll go more and more complex and deeper down the rabbit hole. So given that, today's video is going to be about coral physiology. A lot of people wanted to see other videos based on a poll I did on Facebook, but this is the root of everything, so I figured it's a really good base to start building from. So I'll begin with the anatomy of corals, the anatomy and life cycle of zooxanthella that live inside of them, since they're a very crucial part of coral's physiology. And then lastly, I'll wrap up with components of this coral skeleton. And with that, we'll get started. All right, we're going to start off with scientific naming and taxonomy. So here's a general scheme of how scientists and the scientific community break down categorizing and naming all species on the planet. So first, you have to determine, do I have something alive? And there's lots of different qualifiers for that, but some of the common ones you might know from biology class or just people talking about it are has to be able to grow and divide on its own, has to be able to maintain homeostasis, and it needs to be able to reproduce without a host. So viruses are not technically alive, which is kind of weird to think about. So after you determine, yep, it's alive, you then can classify it into a domain, like if it's single-celled, multi-celled, things like that. Then you start getting into kingdom, phylum, class, order, family, genus, and species. So for example, humans are Homo sapien. That's the genus and species. Let's talk about coral though. Let's say we want to classify and name this branching hammer coral. So we go to our domain, which is eukarya. It's a multicellular organism. It's a eukaryote, so that makes sense. Kingdom is Animalia, so it's an animal. Also makes sense. Its phylum is Cnidaria, so here's a scheme of the entire phylum there. So other relatives in the phylum are jellyfish, the cubozoas, and then also there's hydroids, the hydrozoas. The subphylum that coral as well as anemones belong to are anthozoas, though. Then we have the class, which is Hexachloralia, which are the hexacorals. And then we have the order, which is Seclitaria, which is any stony coral. Then we get into the family, which is Euphyllidae. So you've got things like torches, octos, frog spawn, hammers, all the wall variations of those. Then the genus is Fimbrophilia, which has been recently changed from Euphyllia. It used to be Euphyllia paraanchora, but it's different now. And then you have species, paraanchora, or branching hammer. So that's kind of how you break down and name anything in terms of taxonomy. Now, in terms of coral, this is changing a lot. Things like how Micromussa were differentiated from the typical acan lords. It's like Economensis now, I believe. Or branching lobos are now called Acanthastria pachycepta. There's the Acanthastria maximas. There's lots of different reclassifications that are occurring. And that's simply because A, corals are a really understudied field without a lot of funding, and B, genetics technology is becoming more affordable and is really advanced in recent years. So now scientists are able to take genetics from all these different corals and extract their DNA and classify them using some types of bioinformatic tools to determine, their families and who they're actually related to based on genetic similarity rather than how close they look in terms of skeletal structure or appearance. So now let's get into the anatomy of corals. We'll start really basic. So we have our coral polyp. So there's three primary components to a polyp. You have the tentacles, which sting and capture food. Then those tentacles can bring food particles to the oral cavity. So I say cavity because it's both a mouth and it, passes waste from it. So it's kind of an all-in-one thing. Then you have something of a rudimentary stomach, and then you have the skin, so to speak. So the outside tissue and the inside tissue here. And then sometimes you can have a skeleton depending on the coral. So that's kind of a very, very brief overview of the anatomy of a coral. And starting from here, we'll start to go into the more technical terminology and breakdown of this basic big picture view. All right, now we're going to start getting into the weeds a little bit. So as you can see here, I've kind of labeled each slide by if it's more beginner friendly or it starts to go up in intensity. So this is going to have a little bit more technical application. So we were talking about the skin last time. The truth is there's multiple skin layers or tissue layers, and each of those has a different name as well as a function. So let's break down the external skin. This is technically called the ectoderm. So here's our polyp here. All of this tissue on the outside of the polyp is the ectoderm. This is similar to the epidermis in terms of a human. Now on the inside we have kind of this gastrointestinal cavity. It's like a rudimentary stomach. And the tissue that lines the inside of here is the endoderm. So ecto outside, endo inside, derm just denotes tissue layer. So it's called the endoderm in basically every other species, but because the entire inside of a coral is essentially its stomach, this is kind of called the gastroderm. So you'll see these things be used interchangeably, the gastroderm as well as the endoderm. But just know that basically if I were to say either of those words, I'm just referring to the tissue on the inside of the coral and basically its stomach, whereas the ectoderm is the tissue on the outside of the coral. Now this yellow part here is an intermediate tissue which kind of cushions the two of them. This is called the mesoglia. So mesoglia is very jelly-like, it's very gelatinous. It gives the coral the ability to inflate and deflate and also be very flexible. So if you go back to the beginning slide where we talked about how corals are related to cnidaria, and they're a cnidarian and they're related to jellyfish, if you think about how a jellyfish flows and is basically almost one with the water, it's very gooey if you were to pick one up like a comb jelly on the beach or something, that's because of this high percent composition of mesoglia. So basically, if you're thinking of a coral, think of them very similar in structure to a jellyfish. Same thing here, there are the tentacles, and at the ends of each of these tentacles, there are nemecysts. So nemecysts are essentially like a tiny harpoon filled with venom, sometimes neurotoxins, sometimes, less potent ones. It really depends on the species, how toxic or how potent or how strong the sting is. But they're essentially like a really, really tiny harpoon that's packaged into the ends of these tips. So every single little tip here is loaded with millions of them. And they're almost spring-loaded. There's a very rudimentary mechanism that uses muscles that kind of pull it. And it's almost like a slingshot. So when something triggers that, it just lets go, and the elastic energy lets it out, and it actually punctures tissue. So when corals sting, they really sting. All right, so here is where things get advanced. So not only am I throwing coral anatomy on you, I'm also going to be throwing the anatomy of zooxanthellae on you. So zooxanthellae, for those of you that don't know, or for a brief refresher, are a symbiotic dinoflagellate, basically a healthy algae that can live within the coral's tissue. So they specifically live in the gastroderm, inside of the inner layer of the coral's basically digestive tract. And within that layer, they're protected, but they can still receive light because most coral tissue is translucent. That allows them the ability to reproduce without any form of predators that occur, and then also, well, for the most part, and then also they can photosynthesize. So then they pass on some of their excess sugars that they don't need to survive to the coral. So this allows for coral, or at least most coral species, to be photosynthetic. So corals in reality are an ecosystem in and of themselves. They harbor many types of beneficial bacteria, beneficial fungi, even beneficial viruses. And they also harbor these beneficial dinoflagellants, which make up around 80% of their overall metabolism, at least for the ones that utilize them. So, going into the actual structure of the zooxanthellae themselves, they're essentially just a single-celled organism. So this is a really nice kind of breakdown here. This is from an article from Dana Riddle, so also another really good person of the hobby that's tried to bring science to the hobby for several years now. But basically, what each of these is, some of these are unique. Most of them are, in every cell. So you have the nucleus, which is where all the DNA is stored. You have a vacuole. That can be something of like a chest or like a vault. It's a storage facility. So water can be here, nutrients can be here. The cell can use it interchangeably for the most part, unless there's specific vacuoles, which typically there is, but this does not cover that. Then you have the nuclear membrane, which is a clear coating that goes around the nucleus to help protect it. This also has its own membrane, such as the membrane out here, which can allow it to let certain things in and block certain things out. So it's selectively permeable, which allows for DNA to go in and out when the cell is replicating and things like that. You have lysosomes, which are a type of basically enzyme, which can break down and degrade things in the cell, excess waste. They're like the janitors of the cell. Then you have chloroplast and the thiolate bodies. So both of these together are both very important and are the core components of photosynthesis. These are a subcomponent of the chloroplast. They're a special, basically, organ, or called an organelle, which converts photons or light to energy inside of the chloroplast itself. And then external to this, the chloroplast basically just packages up all the sugars produced by the thiolate. So that's kind of the general structure of zooxanthellae. Next, we'll go into their life cycle. All right, and now you have the life cycle of zooxanthellae. So they're kind of weird because you think of them as relatively stationary within inside the coral. They're kind of encapsulated inside of that gastroderm and enclosed by the coral cells. But in reality, they actually have a juvenile stage that's motile, meaning they can swim around. So if you look at zooxanthellae under the microscope, you'll see these stationary clumps that just kind of float around very slowly. You'll see these crazy ones that are swimming in circles that are just a single cell. They don't really have any direction, their locomotion, no responses they really respond to. So they just kind of swim around. They're very, very weird to observe. The first time I looked at them, I thought that there was a contaminant within the culture, but this is just how they are. So you'll have these progeny here that are motile, and then eventually they can kind of go into a very rudimentary early cell. Then within that, they divide. Then they go into two of the progeny, and then after that you can have them pop into the more advanced cell, which then divides into four and produces four of the progeny again. So this is from a paper. I couldn't actually get access to the original paper because paywalls in science are really expensive. I don't have access to all the journals. So there's my Wikipedia picture. But basically what they were doing was trying to have the whole life cycle of zooxanthellae described. And these gray arrows here with direct observations they made under the microscope. So, they saw this, they saw all of these structures except for this hypothetical one here. So these ones here were kind of inferred because you couldn't have these structures without a structure here that could have divided into it. So this is a rudimentary and theoretical model. There are other models that exist that I think have now been proven and have had direct observation for all steps. But yeah, this word here, symbiodinium, that's just another word for zooxanthellae. It's kind of encapsulates all of zooxanthellae. Going back to the previous classification, that's what all of zooxanthellae are categorized under. So if you look at the kind of root word it has symbiosis or symbiotic kind of in it, which encapsulates what zooxanthellae are. Now this is a very, very fun slide, and will hopefully answer a lot of questions that you might have about zooxanthellae, and that a lot of people in the past have asked me about zooxanthellae. So this is about clade shuffling. The shuffling will be more the next slide, but let's focus on this clade thing here. So here is our whole order of Symbiodinium, and clades are kind of another thing very similar to genus, species, etc. that we went over in the beginning to basically break down and organize an entire order or class of animals. So here we can see that each of these different clades, they're all genetically distinct. Here's kind of where the common ancestor was, and then some of them came and bridged off and are distinct, have different traits and characteristics, different genetics. They're all different species and even different genuses. So we can go here and see that some of these clades can be in hydroids, some in corallomorphs, so mushrooms. You have some that go in zoanthids, some that go in secretarians or stony corals, but you don't see one that goes in every single one of these. And also, it might not even, there's a lot of new papers from 2022 to 2023 that have broken down the individual host and kind of categorized the zooxanthellae in a lot of new coral species. And it's highly, highly specific. So all these zooxanthellae, like add zooxanthellae to your tank, prevent bleaching brands, like unless they have multiple species and they have a lot of these generalists, they're really not going to do much, and they're more of a snake oil type product. They probably have live zooxanthellae, but the likelihood that that zooxanthellae that you add is going to fix X bleached coral in your tank is very, very unlikely. So more so how it works realistically is, let's say we have an acro. Within that acro, if we were to break down all the zooxanthellae within that species, let's say it's a millipora, so a millie, like Jason Fox solar flare, something cool like that. So if we break down all of the zooxanthellae species in our Jason Fox solar flare, we might have 2-3 clades, maybe like a species from, clade C, clade B, and clade F. But those individual species might be different even in Acropora speciosa or a tenuis or something like that. They might be the same clades, but they might be a different species from that clade. And because of that specificity, and because of how niche and different every single coral's gastroderm is, where the zooxanthellae live, these things aren't really interchangeable. Now, there is some evidence, which I'll go over next, that some zooxanthellae can go into different species, but for the most part, it seems to be highly specific. There are generalists, so like you can see here, clade C for the most part can be found in, these are mollusks, like, photosynthetic clams and things like that. So this can be found in a multitude of things, but it's not found in absolutely everything. So the question is there, are there subgeneralists? Like, is there a zooxanthellae strain that could go into every acro? Or is there a zooxanthellae strain that could go in both acros as well as torches and hammers and zoanthids? And the answer to that question is there's not really enough data yet. So I've been in contact with someone that ran one of the largest zooxanthellae libraries in the world for several years. And even then, if you look at their entire database, it's somewhat limited. I believe Dana Riddle has a page somewhere where he categorized every single zooxanthellae that he could find and the host it was in. And if you really look at it, like almost every coral in the hobby has not even been touched yet. So people are not really culturing these or describing them. So this is a relatively new area of the hobby. But the TLDR is most of the zooxanthellae products, probably all the zooxanthellae products, are not going to do much except for act as phyto or filter feeding, food. So here's the data I was talking about that illustrates that sometimes corals can pick up different zooxanthellae, even ones that typically they don't have inside of their holobiome, like their gastroderm and that whole ecosystem. So this was a study that was done with bleached corals. So you had healthy corals, then you had them that got kind of pale and then eventually fully bleached. And basically each of these different little patterns here represents a different zooxanthellae clade and subclade. So D, C2, a different species from C2. And over time, as corals were bleached, you had some of these more thermotolerant or zooxanthella species that could persist and live in higher temperatures that were just kind of naturally floating around the water column. And eventually the coral was able to uptake them. A small percentage of the corals on the reef that experienced the bleaching event were. And then over time, over basically, what is it, February to April. So, but that's a whole year though. So over a year and a couple months period, you had coral that survived and rebounded from bleaching, which then had an entire different zooxanthella composition. So you can see it really changed. There's the initial type. It stayed the same here, but it shifted over time. So this is a relatively large sample size too, I believe, which shows that there could be, a little bit something to clade shuffling, the ability for corals to pick up and exchange different ones. There's also a lot of evidence, albeit anecdotal, in the hobby. So lots of claims of neighboring coral, like you'll have a bunch of acants together, and one was green and one was red, and all of a sudden the red one has a little spotch of green now. Or I've grown torches before, and I have, throw some of the really, really bright Malaysians, like the gods and stuff like that, next to some of the Indo-golds. And it's almost like the Indo-golds get a little bit brighter in 6 months' time, which, there's no controls on that. These are just claims. But it is something interesting where there's quite a lot of people, if you look on Reef to Reef, Reef Central, things like that over the years, that have kind of suggested or at least seen potential evidence of clade shuffling taking place and even leading to shuffling and pigmentation occurring, which is quite interesting. So this is a phenomenon that's at least been documented and would be something that goes along with kind of the last slide. That it would be a really good area of study to cover. So we covered the tissue side and kind of the flesh side of the anatomy, but skeleton is also a very, very big part of anatomy, especially for stony corals. So we'll start off with the septa. So septa, if you're not familiar with them, are kind of like the little teeth. So if you've ever had like a torch die, or if you've had like an acantho that's got sick and you see those kind of spiky edges, it'll look something like this. So you'll have the mouth here, then you have these things that kind of jut up that are spiky. This is like if you were to cut it in half. So these septa are basically parts of the skeleton that have grown that subdivide the coral. And they're different for almost every species and even, or genuses and even species of corals. So septa for a very long time and still now are used as a way to taxonomize corals based on morphology alone. So you can count the number of septa. The septa can have different patterns. There can even be really, really large differences. So like you can have something like how the septa are almost like encapsulated and closed off with something like a candy cane coral versus the open septa that faces outward with Euphyllia or an acantho. So it's really, really different among almost all species. There's really deep septal blades with things like bubble corals, fox corals. So there's a lot of divergence there. And because of that, and because it's so different, it can be used as a useful tool to differentiate coral species. I mean, if you look at the skeleton of a lot of things, you can determine what they are. So based not just the septa, that's kind of below where the tissue is, but you also have the corallites. So if you're looking at something like a Cyphastria, a Favia, Favites, things like that, you actually have the shape of the polyp itself and the surrounding wall. And those can be different shapes, or they can even grow together. They can be space between the cells. They can be back to back. They might only be one giant cell that bleeds into each other, kind of like with Symphilias and things like that. And then another important part is the septum and the basal plate. So this is kind of like the area, like you have the beginning top, like tip-top peaks of the septa, then kind of like the flat plane there. So it's kind of an intermediate, really thin skeletal layer between the tissue and then the coral itself. So that's kind of a good summation of the skeleton side of things. And I could certainly go much deeper on this in later videos, but most of my other content will be more pertinent to the tissue side of things. So if people are interested in deep diving on the skeleton, I can go really in depth in the skeleton, but I figured I'd do more bird's eye view here. So here's kind of the granddaddy chart. This is a good summation of everything we've covered today, as well as more that I'll hopefully cover in future videos. So as a, an end here, we've got our mucosal lining and our ectoderm, our external tissue layer. We've got our tentacles here, which have nemesis on them, our stinging cells. You can see that broken down right here. Then you have the oral cavity, which goes into the gastroderm. There's a breakdown here of that tissue. So you have the mucus, the ectoderm, the mesoglia, that kind of really rubbery, gelatin-like tissue or intermediate tissue that serves as a rudimentary form of the mesoderm that are in more complex organisms. Typically, there's 3 tissue layers, only 2 in coral. Then you've got the gastrodermis or the endoderm, where the zooxanthella are housed. Here's a better breakdown of that. And then you go down to the skeleton, where you've got the septa, the basal plate that we touched on briefly. And then I didn't really talk about this area, but I could certainly do it in future videos. I plan to have some on coral spawning and reproduction. This is kind of the business side of the corals. This is the gonads, where there's all the sexual activities. So there's egg and sperm production, as well as the primary oocytes. I can go into development on that if people would like. And then here are my sources if anyone would like to visit them and read these full articles. Everything was essentially from a peer-reviewed study, besides the couple things I had to take from Wikipedia because of paywalls, and because also there's pretty good diagrams on there sometimes. But if anyone has any questions, you can contact me on Facebook, leave a comment down below, and I'll get back to you as soon as I can. I really appreciate any support. This is my first YouTube video, so it might be a little bit rough getting familiar with editing and things like that, but hopefully it wasn't too choppy for everyone. I'm open to any criticism. Let me hear it. I really would like to improve this content and deliver to all of you. Leave me suggestions for content in the future. I'm probably going to be talking about, the microbiology of coral, so going into the beneficial partners, the pathogenic, things like that more so in the future. But I'm open to water chemistry or doing things like that as well. And please like and subscribe. I'd like to make some more content. And if you hit the notification bell, you'll be notified on when I upload. Thanks, and see you later.